Hello and welcome everyone in another episode of Research as Celebrity. Today we have Dr. Ajit Sharma with us. A brief introduction of Dr. Ajit. He has done his bachelor's in microbiology from Punjab University, Chandigarh, India. And then for master's, he joined Jivaji University, where he did biochemistry. And for PhD, he joined Homi Bhabha National Institute in Mumbai and he earned his degree of PhD in life sciences. Then for postdoc, he traveled to Canada and Germany. At Canada, he joined University of Alberta, Edmonton, and in Germany, he was part of Institute of Molecular Biology. Currently, Dr. Ajit is staff scientist at National Cancer Institute and National Institute of Health in the United States of America. With this brief introduction, I would like to welcome Dr. Ajit by saying thank you so much for taking time out for this conversation.
Thank you, Raja, for inviting me for this session. It's really a privilege for me to be here. So basically, let me tell you something about uh, NIH. So I came in US in 2022 October, and I joined NIH as a research fellow. And basically, we are working on a small cell lung cancer. And after serving to NIH as a research fellow for 1 year, then I promoted to staff scientist. So my job is to train post-bac, summer intern, do my own projects. So I have 2 projects I'm handling right now. And then another part of my job is to train medical fellow also. Because we are working on clinical projects and my boss is a he's a clinician, so he look on the patients and whatever we observe in the patient we try to answer in lab. So it's truly and it's privilege to be working in this setup from like bed to bedside.
Nice. So we always start these conversation with this question that how and when you decided that you want to become a researcher.
Okay, so this is a very complex question. I can say there is not a single incident which basically motivate me to be in a science. So the whole credit initially when I born and then my mother bring me. So the main credit goes to my mother. Although she was not so educated, she is just 10th grade pass and then but she has a great vision. So she wanted to me as a doctor to become a doctor when I will grow up. So what she used to do that when I was 3 year old or 4 year old, so she brings some toys for related to human body, and you have to mismatch or match with organs. So that the idea is to train me and to understand how human body it is. So that generate a lot of curiosity, and she brought some like toys like a stethoscope and all. So she made like environment so that I can think about medicine and the human life or science. Then I went to school. So I was very fortunate when I was in ninth standard. So we have a science teacher, Dr. Bhattacharya. So she basically inculcated habit of thinking about science. So she was teaching us like atom, electron, proton, neutron. So that actually elicit me to think about science, to learn about science, and to read about science. Then when I went for high school, so I got a wonderful teacher. So because teacher, contribute a lot. If you get a good teacher, he can or she can inspire you whatever he or she can induce or interest in certain things. So when I was in 11th grade, so we got a nice teacher and she started teaching us genetics, cell biology and all. So that basically drive me to think about science, to learn about science. And the overall the environment in my house is they always push me or motivate me to become a doctor. Unfortunately, I didn't able to clear the medical exam. Then I felt deserted. Then I went for like undergrad program. So in Punjab University, microbiology department. But it didn't work well because I didn't get a very nice teacher. Although the university was very big, but anyhow, means it's a general curiosity to learn about the science. So I keep far in myself. Then I went for master program in Geology University, Gwalior. There I got a wonderful teacher. Like there's couple of teachers I want to mention them and I want to like acknowledge them. So we got a teacher like Dr. Y.K. Jaswal, uh, Purnima ma'am. So they teach so nice. And I think one day I found that I was just roaming around the university. One day I found that Dr. Y.K. Jaswal, so he put a lot of effort in preparing his lectures before he teach. So I found that he is making like the like what he has to teach. So it was 11 o'clock. So I felt like our teachers put so much effort in teaching us, and the way he is to teach, he is always welcome. Like he doesn't teach by hour. So after class, he stopped by. We have a lot of questions and curiosity. So he used to answer, and he never felt like tired. Okay, I'm done and I have to go. So as much as we can answer, he is ready to like as much as we ask questions, he is ready to answer for that. Or if he don't have answer, he go and read and then class he is to answer. Then we have a Pune ma'am. One day construction was going on in our department. She was literally sitting in the car and preparing the like for the class what she has to teach. So if you get such a nice teacher and they teach science as a science, they don't teach science as a history. So they always teach like you are forced to think. Thinking is very important when you read science, right? So and then their question is not like you mug up and like memory based, not a question. They always give a question where there is a scope of maximum thinking. So like mugging up doesn't work at all. Uh, so they actually shaped me in like taking the journey of science. Then after finishing my master, then during the master program, we have a 6 months like training program. You can go to some national institute and you work there and learn about the science. So I went to DPAS in New Delhi. It's a defense institute where they do some physiological based research, but that was not my interest. So like I managed. I stayed there for 6 months, and like I'm very vocal about what what is true and what is thing. I don't want to manipulate. So I went there, but that training doesn't help me in thinking, and basically it's a herbal based project. So I did it. I have to finish it. But those 2 years master program, like the teacher what they taught us about cancer or cell cycle and all, that actually keep fire in me to opt for science to do PhD. And another reason is to do PhD is I promised my mother that anyhow I cannot able to become a doctor, but I will add doctor to my name. So that is the two basically motivation to opt for science and to do PhD. Then after that I joined SGPGI for a short time. So I joined a lab, medical genetics lab. So I was exploring thalassemia there for a couple of months. In between, I got like application for PhD program in Tata Cancer Center in Mumbai, and which is always I wanted to be there to learn about cancer or cell cycle. So then I applied, and fortunately, I went through written test and interview, and then I was selected. So there were 10 seats, and I was selected there. So the environment in Tata Memorial Cancer Center is very nice. So they adapt international, like a international program. So we have 1 year coursework where they teach cancer biology, and you have to qualify uh, all the time. You have a exam, you have to present papers, you have to understand paper. You don't you don't only have to present the paper, but you have to criticize the paper, what is lacking in the paper, which is not so reasonable because when you read some paper like Nature or Cell or something big paper, so there is a less scope of criticism, but they want to actually test your natural way of thinking. That's the idea behind how you criticize the paper.
Mhm.
So you have to score for, they give you a score, and then after finishing coursework, so you have to score minimum 60%, and then you are able to register for the PhD program in Homi Bhabha National Institute, which is called as HBNI. So HBNI is a Department of Atomic Energy program where all the institute which is registered under DAE or comes under DAE, so they make a university, deemed university, which they named as Homi Bhabha National Institute. So we registered there as a PhD program in life sciences. So after end of finishing the coursework, then you have to decide the lab. This is the biggest challenge. Like if you, and what I believe that if you, the perfect fit, that you should always look for a mentor who can be an ideal mentor, who can, and mentor is the source of motivation. Like for two things, mentor and the subject of your research, which always motivate you for the next 4 or 5 years during your PhD journey. So I was very clear that I want to choose a area which is very global, not very specialized. So if I have to understand something in future, I can able to do that. So I choose a area, and at that time in 2007, chromatin is the like very hot. So everybody want to understand histone code or histone biology, how histone code regulate the whole physiology of the cells, cellular physiology. So if somebody learned about the chromatin, he can do plant science or plant molecular biology, plant physiology, biology, anything you can do. It depends on the question what you opt for. So I choose a lab in Tata Memorial Cancer Center. Dr. Sanjay Gupta, who was working on chromatin field. So he is trying to understand role of chromatin in gastric cancer. So then I joined it, and then you have to present paper after a couple of months, and they will grade you, and then finally you get registered. So then you have to choose the topic, what your research will be for the next 5 6 years. So at that time, people were trying to explore the role of chromatin to understand how chromatin play a role in DNA repair or genome instability. So my mentor told me that we don't do this. We just understand the transcription of chromatin and gastric cancer. So I have no idea about DNA repair or genome stability. So make sure that if you are choosing this field, so you have to read. And of course, I will also explore because this is something challenging for me to explore it. So we decided, okay, I will go with DNA repair and chromatin, and that's very exciting because a lot of big paper and a lot of nice work is coming out. So then I started exploring the histones and histone code. and role of histone code in DNA repair and genome stability. So I developed a project where I was trying to understand the role of histone code in different cell cycle in context to DNA repair. So I developed all the methodology and the project by myself. It's very tough work because I was dealing with the cell cycle and it's very time bound. So like the radiation source is very far away and then a lot of patients are there. So you have to manage the time. So anyhow, I get up at 4 o'clock morning and then you have to block the cells in a particular cell cycle, release them, go for radiation, come back, and the experiment, the nature of the experiment is very complex because you have to do the DNA repair kinetics to understand whether when you induce the DNA damage, how it recover, then cell cycle, then you have to isolate the histone. So multiple things you have to do. But I was very, very, lucky in that sense that my mentor was very supportive and I learned the technique how to do the things in a multiple task in a same time. So after juggling with project for 2 years, so it's very difficult to find something new. So, but which I always believe and which I always tell people that if you are in a science, there is only one thing which keep you alive in the science. That's a curiosity to know your result. So your result basically drive you throughout the science, what you do. There is nothing like most of the thing is very challenging. There's a like most of the experiment you do, it fails. But are you ready to take challenge next day with the same energy, same pace, same motivation? And if it fails, then are you able to keep doing it until you get succeed? This is the major thing what new generation and people should and I'm learning day by day also that how I how I should be keep alive to 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 actually for the curiosity to know the result. So after juggling for 2 years, because the cell cycle, how it works is histone changes, modifications. So in G1 phase, S and G2 phase, histone code is very different, and it's very difficult to find very specific change in context to DNA repair. So still, I remember that I was developing some blot, and at night, almost 11 o'clock, something came, and it's a very peculiar change, and it changes in context to some DNA repair marker. So I didn't sleep on that night. So I run again, same experiment to know whether this result came by fault or there is some mistake while I'm doing it. So I run again, and then in the morning I got a same result, and then I went for like to my hostel and take a like nap or sleep. And then I realized that okay, this is a result which I'm going to chase for next 4 5 years. And then I was sure that okay, now I can develop the project. I have a specific questions. So in science, actually, nowadays people run for learning the technique, which I always say that technique is not important. Your question is very important. If you have a question, you can learn the technique. You can like depend on your like questions. You can develop the technique to address that questions. So then I was so happy that I got some questions to address, and then I like it was a very smooth journey after after 2 years of juggling, and my boss was very happy that we proposed something, and that was something very new to find the histone modification in close contact to DNA repair in a specific phase of the cell cycle. So and then after that, I found the signaling pathway. So the overall my training during my PhD is chromatin and hardcore biochemistry and molecular biology. And then we found some signaling pathway which was regulating the that DNA histone modification in close to DNA repair. When we affect that signaling pathway, then cell become very sensitive. So that is the thing. Then when I joined Tata Memorial Center, so I realized that whatever I do for next 4 5 years, that will not directly or in a very short span of time will benefit for the people. So I planted 2 mango trees. So that's another motivation. Like I keep growing them, putting some urea to grow them. So when they grow, I feel that okay, I'm also growing. So
Mhm.
that's a part of like I connect with that, and then my project was growing, the plant was growing. Finally, I finished my PhD, and then during acknowledgement, I dedicated those to plant. And before my defense, I felt very happy to see that some patients were taking lunch
Mhm.
in the shadow of the plant and they were taking rest. So that gave me some satisfaction that okay, I addressed some questions during my PhD program, and then this is also direct effect, a benefit for the patients also. So I acknowledge those 2 mango tree to my institute. Then after that, I was looking for my postdoctoral training. So the idea
Yeah.
is to look
No, so
for
I think I'll just take a break here before we transition into the postdoctoral journey. The things which you have mentioned are, I think, is universal for all the researchers, the people who
yeah,
has done their PhD a decade ago, or the students who will be joining PhD after a decade. One thing
yeah,
which remains the same are the struggles, the failed experiments, the motivation which, as you mentioned, that keeps always you like, okay, are you ready to face this challenge today?
yeah,
Because the experiment has been failing for a day, a week, or sometimes even more, that you should be motivated about it, and every day morning you are ready to face or to set up the the experiment by troubleshooting various parameters, because one thing in science is it should work the way it should.
yes,
Either we have to tweak or we are missing some part. That's why the result is not happening. And
yes.
you mentioned cell cycle in a nice way, that there is no way that a cell jumps from one cell to another. Yes,
It
there are
has to go from one phase to
situations.
another.
Yes, exactly. There are biological rules which needs to be followed, and that is what brings a lot of challenges. But what I want to ask your thoughts and how you will suggest or guide our current PhD students, that when they face these challenges, how they should approach it. And you mentioned great example of planting a tree, because now
Yeah,
you have this motivation by looking at them, that okay, they are growing, my project is growing, I'm able to help uh, patients in real time, because the
yeah,
research might take some time from bench to market and then
bedside.
help them.
Yeah.
Yeah. So what are your suggestions and guidance for the researchers, either graduate students like PhD students or even postdocs, how they should keep their head up?
Okay. So what I realized while like I was very fortunate when I was doing PhD. So my mentor always encouraged us not to use any kit-based essay. So he always believed in very crude biochemistry, crude molecular biology, and always use manual methods. So when you use manual methods, you know each and every ingredient, what you are going to add, why you using it. So nowadays, new generation, when I went to postdoc and then I'm working in an edge. So people nowadays want very things fast, and that's a sometimes the experiment fail and you don't know how why it failed, because you are using kit, and kit is not going to disclose their ingredient, their buffer. So then you develop less like knowledge about the technique and the assay. So it's very hard for the difficult for to troubleshoot. So I always motivate, like when I joined NIH, so we have a culture to get a like a ready-made gel or all the assay, ready-made assay kit. So I develop in my lab to use all this manual technique to cast a gel. So whenever any post back or summer intern come, they have to learn, they have to read a lot about the technique, crude way of doing it. And slowly they learn, and then they realize that this helping a lot. When experiment fail, they can able to independently troubleshoot, and they come up with some idea that okay, this is the problem why the AC didn't work. So I always motivate everyone to use manual and crude way before you do any experiment. Take time, read lot about the experiment, about the background of the experiment, so that you know everything about the essay. If something felt like most of the, like I always say that when we do 100% work in research, 95% goes in dustbin. Only 5% whatever we see in the form of publication, it comes as a result and what we know. So multiple failure happened, and each failure teach something. So whenever any post back or summer intern or postdoc come to our lab, I always say that mistake is welcome. You can make thousands of mistakes, but don't make same mistake thousand times. The reason why I say that when you make thousands of different mistakes, with each mistake you learn something. But when you are doing same mistake thousands of times, you are not learning anything. So that's a wastage of resource, time, energy, and everything. So these are the two, three things which I always encourage any new people who are joining PhD. Read as much as now reading is missing. Like everything is online now. So last time we had one very nice student. She came as a summer intern, and like in US or Canada or Germany, I don't see kids are reading on like notebook or getting print. So there is always a scope when you print something, you read, you highlight, you write your comment, what you have understand. So her note was very colorful. I felt so impressed that someone I found that reading on like printing literally and reading. So I asked her that I found something very unusual that you are reading, you don't read on your laptop. So she said, no, my mom, dad is also a scientist, and they believe in like printing and reading as much as I can. So that gives us habit of reading. Like when you read on laptop, half an hour you feel very tired. Then you sit down, and then there is a less chances of reading. So I always encourage as much as people should read before do anything, or if somebody is opting for any research field, please do literature survey and everything. Second is use manual method as much as you can. It takes time, I understand, because now it's very horse race. Everybody want to get as maximum as in a very short time, but don't take shortcut. Take manual method, use as much as manual techniques. You learn better. And third thing is like research is something, it's not like a 1 day or 2 day job or like you will get maximum. So it's like a continuous learning process. So keep motivation up all the time. That something it failed, you once would realize that if something failed, you will learn something, and then you come up with some successful result. That's fine. But I think success and failure is equally important in research.
Yeah.
So
Absolutely. And I think the points which you mentioned here, I would just like to say to all the audience. So this is the universal for every researcher at any stage of your career. That as Dr. Ajit mentioned about reading. So if you are a new researcher joining PhD, okay? So for your review of literature, to understand the topic of your interest, reading is non-negotiable. The more you read, the more up to date you are with your review of literature about the current status and what has, from where the field has started. This entire thing will prepare you to start thinking that what has been done, what needs to be done, and that's when you bring out your research question. Then the second point, which I would like to emphasize on for everyone here, kits versus manual methods or so. We often listen or hear this from all around the globe that this kit is not working, this process is not working. People have spent weeks and sometimes even months, and repeatedly it is happening. What you can do is you can tell the company, and then they can provide you another kit as a replacement. So money is not lost there physically, but what has been lost is the resources, your time, and the most important thing is the mental health. You think it is not working because you are missing something. It might be that one of the ingredients of the kit was not either well stored in the lab or
yeah,
had some manufacturing issues. You can never troubleshoot it until unless you know all of it, as Dr. Ajit said, that if you know how to do it manually, you can always do it, and then you're like, okay, now it is working here. All the failed experiments comes at a cost of mental health. The
yes,
stress it brings, and it makes it even more problematic for everyday's challenge, as Dr. Ajit mentioned, that when in the morning, are you ready to take this challenge on seventh day, 10th day, 15th day, or even second month? Like, because you are doing everything as per your book. You are following everything and still it is not working just because, as we mentioned, it might be one of those problems. And third, obviously, is the research is the continuous process. You might have learned something, you might have discovered something, but now taking from bench to market is the real journey of any researcher. And this keeps on going on and on and on. You might have 10 products already in the market, but now sometimes just cutting the cost of the product, making it more reasonable, more stable. That's what we keep on working, and that's why research is that vicious cycle. Once you can only enter it, you can never exit. You can change academia and industry, but you can never go out of research ever
yeah,
because once a researcher, always a researcher. This is something which I always say. But now taking next step, Dr. Ajit, after this PhD, the postdocs, 6 years in Canada, 6 months in Germany, now in United States. What will you tell about the various challenges outside research, what you have faced, and how you can, suggest or guide our researchers who are planning to change their countries? Either it is from India to somewhere else or globally, because this is something which we have seen that researchers often change the countries.
okay. So I want to tell something very important during when you transition from one country to another. So like when you move from PhD to postdoc program, so the country changes, culture changes, the way you do things, a lot of things changes. Like each lab has their own culture of working. Here you are working in a in a domestic setup where like a lot of people are from same cultural background, or they understand your language or accent or something like that. So when you move from PhD to postdoc program, then one thing is finance is very important. So I always encourage people who are doing PhD to save some money. Don't put burden on your parents. Um, like government of India, in context to our country, government of India is paying fairly and good enough, uh, like a stipend. So one should save some money, and that will help you in applying for visa and then getting a like air ticket, and then move. You need some expenditure for 1 or 2 months there, because directly when you go, you will not get a salary. So, and then you have to spend some money on clothing and your preparation and everything. So, one thing that please try to save some money while doing PhD. Don't put burden on your parents. If you are, since I'm belong from middle class family, so everybody can understand the challenges of middle class family. Second thing is the real challenge when I moved from India to Canada is I was doing PhD in Mumbai, where the temperature is very Mediterranean, like not so cold, hot. And our institute was located in Navi Mumbai, so we get a very fresh air all the time. Windows were open. Uh, when you move from India to Canada, so when I moved from India to Canada, so I went to Alberta, which is extremely cold. Like I always say that it's a crude cold. So I remember I get down there in September, and after 15 days, the snow started. So I was very excited, and I was just dancing and singing, because first time we see, right, we don't see, we see in the movies, or sometimes if you go to Shimla or something, you see this. So my landlord told me, don't get excited, you'll get all the time this. So like then I joined a lab, and then the lab was very international. I was very fortunate. All the time I got a very nice mentor during my postdoc program. My mentor was Dr. Michael Hengel. Is very popular and very well known in chromatin field and quantitative microscopy. He is the guy who first developed the colored electron microscopy. So he incorporated colored for the protein, DNA, RNA. Very, very extremely highly intellectual, very like very high level of IQ level. So he told me, see Ajit, I'm not here to spoon feed you. And he is also very classical scientist. So he don't also believe in kit best techniques. So he trained in 80s or 70s. So he did, and he has a unique combination. So he did his PhD in with a very big guy in chromatin, and he learned biochemistry. Then he went for his postdoc program. So he has a microscopy field, so cell biology. So it's a deadly combination, which I always say in the field. If somebody has a biochemistry and cell biology combination, it's a very deadly combination, a very unique combination. So he was very intelligent. So he said that I'm not going to spoon feed. I generally trained postdocs, so I give a liberty and freedom to explore the area and come up with some specific questions. Of course, I'm always there. If you trap in certain things, then we can juggle and find the way how to like get rid of it and find a proper technique or proper way of doing it. So this is what, and second thing is I'm going to provide you enough money and lab. So this is what I'm going to do. And it's your life. If you work, you'll excel. If you don't work, after 4 or 5 years, your life will be done. So you have to face the consequences. So it was very clear. And I was I feel very lucky about it, that I got such a very clear mindset mentor. He never asked me when you are coming, when you are going, whether you work during weekend. And the lab environment was very nice, not so toxic, so very helpful people. And although for the first 1 or 2 months I had a challenges because each person has a different accent. So somebody is from Egypt, somebody is from China, somebody is from Canada. So everybody has a different Morocco. So their accent is very different. So it takes time to pick up the accent. And then second thing is when you join the big lab, so it's a no man's land. So you have to explore everything of your own. Because when you ask something, then it's a very competitive environment also. So people will tell you once or twice, but when you keep on asking, then they feel burden also. So that is the second challenge. So slowly I overcome with it. And the way of working in India helps always. We always criticize something that, but I think I took it as like whatever I learned in India that helps me a lot. So during Christmas time, so I just went through all the like inventories in the lab where it is, and because it's a big lab, we have huge resources about cell line. You have to just think about the essay or some question, and in all the resources are there. So you have to just find the question and way of doing it. Outside lab, I think the biggest challenge is weather. Very cold. Like it's very difficult. Like at that time, I didn't have a car, so I used to walk also in such a extreme cold. Sometimes I used to take bus, but there is no any physical activities like. So I thought that I should walk so that I can be a fit. But later on, I realized that's not a good idea to walk in a cold. So the temperature goes like -30, -40. Uh, so, and it's very extreme cold, and all the time you see just white, no green, nothing. So 6 months, so cold start October and it end up till end of April. So all the time you snowing. There is no social activities, no friends. So for the couple of months.
So this is like for all the audience here, that how these things, what you think, you might not even acknowledge that shape. Although
Takes
like
time
when
for me.
these conversation with the some of you researchers who are thinking to move out of India, some of you are really intelligent and have seen your way out that okay, what it will be like. As Dr. Ajit mentioned, he was not prepared for those extreme colds. But when he landed there, that's when he realized. Some of you already have done your homework about the climate, about the socio-economic differences, about the cultural differences, because what you can say here in India probably is might be offensive in some other part of the world. So if you are
Yeah.
prepared for all of this, you can enjoy your research outside for sure. Inside the lab, it is the thing which you have been trained for, but outside the lab, you have a real life also. Okay, so
Yes.
always try to be part of the prepared for that. And with this, we'll say thank you so much, Dr. Ajit, sharing this journey. And I'm sure that this will change life or at least the thinking process of so many researchers after listening to this conversation. We always say that if our audience wants write directly to the researcher celebrities, they can write to you. If you want Empowering Science Foundation to put you in touch with our researcher celebrities for your for their mentorship program or so, you can write to us. The only thing which we want from all the audience is your passion for research. Do whatever makes you happy, and if research happen to be them one of those things, we are here to help you as much as you want it, as much as we can help it. With this, we'll say thank you so much, Dr. Ajit, once again for taking time out and sharing your journey. And for all the audience, stay tuned for another episode of Researcher Celebrity.
Thank you, Raja, and thank you for inviting me, and thank you, your audience. I always welcome for the question. If somebody has it, I will try to answer it. May take time sometimes because of the busy life, but I will happy to answer for sure.
Thank you so much, sir.
Thank you so much.